In this module, we'll talk about patterns of inheritance that don't follow Mendelian genetics. So let's look at the first category, incomplete dominance. In this pattern of inheritance, heterozygotes show an intermediate phenotype, sort of like blending. Example given here is true breeding, white flowers are crossed with true breeding, red flowers. The flowers here are snapdragons. People here might be familiar with them with the name Kutta Phool. Anyways, when these two plants are crossed, the F1 generation is pink. So, although the red is dominant, but it is not completely dominant. It is therefore called incomplete dominance. It is giving the F1 generation of two breeding parents of different genotypes are giving an intermediate phenotype. So, is this blending? It looks like blending, but it is not exactly blending. We can prove that if we take the heterozygotes and we cross them, we mate them, we get the following ratios. So here you can see we have two pink flowers. The ratio is 1:2:1. So we have two pink flowers and one white flower and one red flower. So the gene for the white flower, when the F1 generation made its gametes, was again segregated. It segregated. That is the only reason we were able to get white flowers. Additionally, we can do a test cross, which we have talked about previously, between a heterozygous pink flower from F1 generation right here with the homozygous white flower RR. So we know if we cross heterozygotes with homozygous recessive, 50% of the progeny will be heterozygous and 50% will be homozygous recessive. That's exactly what we have. So 50% of the flowers in a test cross were pink because they were heterozygotes, and 50% of the flowers were white, they were homozygous. Basically, these flowers, they do not follow the complete dominance, therefore it is called the incomplete dominance. Now let's look at another mode in which genetic information is passed from one generation to next. Next, I would like to give you an example of codominance, in which both genes are dominant and they are fully expressed. The classic example of codominance is blood groups. These were identified, blood groups were discovered by Karl Landsteiner in early 1900s. Before that, attempts to transfuse blood from one patient to another were not always successful. As we know today, that blood groups, if we transfuse blood which is not matching, it can have serious consequences for the patients. So he basically took the blood from patients A with the blood group A, and he added the serum. Serum is basically blood without any cells. So if we add plasma from B blood group into A blood, this results in accumulation or clumping of the red blood cells. Similarly, if a person with A blood group is exposed to plasma from B blood group, that will also cause clumping of the red blood cells. This is basically an example of codominance. Both A and B genes are expressed simultaneously in people who have A blood group. They make both these A and B elements and display them on their surface. That results in basically a phenomena called codominance. Next, let me introduce you to another concept, epistasis. It is a phenomenon in which effect of one gene is dependent on presence of one or more modifier genes. For example, in mice, the coat color, the brown coat color is dominant over the black coat color. However, the ability of mice to produce color on their coat is dependent on another gene. If that gene, let's call that A gene. If A gene is present, A gene, capital A, is the dominant phenotype, is a dominant form of the gene, dominant allele. If two capital A's or capital A, small a are present, then the mice will be able to produce color in the coat. If, however, our A gene is recessive homozygous, that is small a, the mice will not be able to produce any color to begin with. So ability of mice to produce color is not residing in just the in the heterozygosity, is not present in just the color gene. It is also dependent on gene that allows these mice to produce color in the first place. So this phenomena is called epistasis. In next module, we'll talk about genetic experiments on flies.